Get started. Have to be out by 1, so we're going to be very punctual because we have teaching events afterwards. So I'm Lars Heckman from the GI division. I'll give you the very brief introduction. 1 minute, I promise. Our speaker today is Dr. Pradeepta Ghosh. Very cliff notes, medical training at Christian Medical College in India. Then she did a postdoc at WashU to work on cell biology and G protein signaling, but she discovered a major group of single modifiers. Then came to UCSD for residency and GI fellowship and joined the faculty, where she's now a full professor in the Department of Medicine, as well as in the Department of Cellular Molecular Medicine. The reason why she's here now to talk today is not about her research primarily, but about the other efforts she's been involved with in starting and supporting a number of cores and centers at UCSD. So she would qualify as a serial academic entrepreneur, if there's such a thing. And so the particular reason we're here now is to talk about the humanoid core, which is dedicated to making human cell culture and sophisticated cell models available for studies, which fits nicely into the NIH priorities recently announced to focus on in vitro models. And the NAMs, the new alternative methodology models. And with that, Pradeepta, you got it.
Thank you, Lars. I was going to tackle him if you didn't finish soon enough, but thank you so much for that gracious introduction. Can everybody hear me well at the back? I am a terrible speaker behind a podium. So for those online joining us today, thank you for being there. And those here, thank you for being there and tolerating me walking around this entire time. With that said, I have a lot to cover today. And as you heard, it is not about what I do, not my lab's work. It is about something that, let me get the lights down. Maybe that's, hopefully that's okay, seen easily. There are lots of microscopy pictures, so that's why I turned it down. So when you look at a disease modeling, you're talking about human disease, our body is a very complex system, extraordinary complexity. And it's so non-intuitive, that spider web that's jiggling around there. I'm pretty sure you missed that 2 nodes never let go this entire time I've had you distracted. And that is perhaps why 90% of the time we try to hit, we miss. And that's about the drug discovery success rate right now. It's 90% failure. So what was the next way how we were going to overcome this limitation? Well, turns out it was supposed to be NAMs. NAMs that are new approach methodologies. They are supposed to be made to be precise models. And it was supposed to be grounded in truth, which is patient, which is cohort level anchored. And somehow it's supposed to have integrated math, computation, all of it in it to make them precise. And yet, NAMs are nothing but these assembloids, cells outside the body, in culture, filled with artifacts, in extracellular matrix that looks nothing like the organs. That on the right-hand side is the criteria that the feds have laid out. It ought to have all of these elements, end-to-end human, integrated with computation into cohorts, population. Phase 3 rider. That is a picture taken by our own Kyle Dykes, right through the microscopes in humanoid. And this is what humanoid is dedicated to. Today, I welcome you to take this journey for the next 45 minutes or so to see what these men and women have accomplished. They are not my lab. They are not part of my group. And yet, don't think they are innocent. I'll tell you quickly, briefly, how well they know me. And this would be the next only minute I'll talk about me, because it's the relationship with these people and what they have accomplished with just me working with them. You see, I have a condition that makes me extremely disciplined and extremely predictable. And only these people know how to take full advantage of it, because they know there's nothing in the world other than telling predictor first thing in the morning, there's something interesting, and then walking away completely for the next 24 hours, because that's exactly how I'm left. And only they can do it, not even my lab. Or getting completely without fully dressed in the restroom because they knew I was going to take a break between my meetings to go to the restroom to just show me data. Folks, that happens. And that's what I needed you to get out of this slide. These are men and women excited to do what they do. And if you see me excited today, it's not to make you feel anything other than just I'm trembling with excitement, because I'm really excited to show you what they have accomplished. That said, this is what Humanoid has done for 6 years. 8,900 vials is not the story. Organoids of 23 diseases is not the story. It's not about 8 different organs head to toe. We don't grow organoids. We just grow the confidence in what they predict, because I am here to show you that what organoids can do has not been realized. We are just beginning to know that. It took a village to get this biobank. It started under the leadership of Cleveland and Davison at CMM. It found its headquarters in the basic science department. Then and now, seamlessly continuing with support that is end to end from that department. But guess what? It's a basic science department. It does not have patients. Here is what made Humanoid. This department, medicine, was the largest contributor to its biobank. So was Pete, surgery, Center for Prenatal Discovery. You can read it. And guess what? It gave it all back. Every time it brought in funding, projects, new excitement, we gave it back. And this is the charity that CMM helps host. And for that, I'm extremely grateful, because something like this had to be a love child between medicine and CMM, and couldn't have been better put together than this framework. And I hope you will recognize that very soon as I move on. But that's not all. Look at the impact for 6 years. Humanoid has served the globe, biotech, industry, trainees from different parts of the world. We have sold not organoids. We have sold a vision for how to use it, how to make it reproducible, cost-effective, conditioned media that you can use and reproduce over and over again to get your results. We were just working on getting the blueprint right. And that's because of these men and women. When you see that, you don't see what the 6 years turnaround was. 9 different careers were launched, but don't you worry. Look what's coming next. Right in that picture are the next assistant professors who are waiting to get recruited. There are graduate students who are waiting to come in. These are the people who Humanoid is launching. And guess what? They all want to stay at UC San Diego. And that is how Humanoid is making sure that if the feds have said, this is what we want to train people in knowing how to use it. But that's not all. Our impact in training was the best way to spread those seeds. And we went beyond the borders. We are right now hosting our first Fulbright scholar right here at Humanoid, and a graduate student from British Columbia. because humanoid uniquely provides that immersive on-site training that could take paperwork, could take a lot of logistics, could take tuition and other things figured out. But thank you that you all were there to figure it out how to do it. And we did it. It offers the training. That's the only way to get everybody ready. But let me take you down the history lane of we were not the first here. This movement of not using animals started in 1981 when human models were not a legitimate science. I connected with Dr. Horton, who directs Johns Hopkins CAT. He will visit us next time he's in San Diego. Look at what their priorities was. It was to reduce, refine, replace animals. But not until Dr. Hans Clevers brought the ability, technological leap, the breakthrough of creating genetically stable, expandable, cryopreservable organoids. Not until then, we could have started what is humanoid today. It was just a dream, a fuzzy dream. We wanted to create a framework for vetting humanness of NAMs as end-to-end clinical translation for reproducibility, relevance, and regulatory readiness. 3 Rs that were not even formulated by the feds at that time. We were just 6 years too early. And not until then, I just did it. Look at that date, September 25, 2025. Not until then did we really have the first ability to do something that would then be adopted by the feds. Because NIH did the only thing they could have done, which is to put together all the regulatory agencies on the same framework so that a workflow you will see today has the hope of getting to the FDA approval pre-IND workflow. That said, it was very important. 6 years ago, when organoids weren't a legitimate science still, industry didn't approve it. Biotech was scared of it. Anybody who has a reasonable familiarity with organoids dismissed it as a ball of cells. That anybody was me also. And there was all the reasons one would get really famous for the wrong reasons. Really famous for the wrong reasons. So many ways there were traps. There is no playbook. There was no playbook. Founders don't have a map. They have to chart the path and sometimes be okay with where we end up. I am here to share that with you today, where we ended up. But it took some thoughtfulness. So let me quickly tell you. I'm incredibly grateful that humanoid in our journey was given that kind of framework by so many right here at UC San Diego. It did take a village to make sure responsible conduct of research with human subjects had the right regulatory support, had the right data science, data infrastructure to maintain a coded data bank, which means every patient was their virtual replica. It meant navigating IP territories which needed OIC to keep us safe, not do stupid things. It needed funding, it needed personnel, it had the support when we needed it. It also needed complex things, telling our stories, getting them right without the unnecessary hype, being transparent, being authentic. And soon enough, we learned there was so much support here from the right people at the right time. And you know all that it took? Simple. We were just We just had to explain what we wanted to do. And we found people naturally came by and supported it. They gave us everything we needed to make it happen and to get it right. They just needed to know the cause. And they stuck with us. This is the most important slide for those of you who want to use maps. An IRB protocol like no other. We were told, go ahead, pick an organ, pick a disease. We couldn't. No, that's not possible. It's head to toe, womb to tomb, any disease, any health, any organ, any tissue, prospective, living, decoded. You have an IRB, you snap, connect, and go. You don't have one, add you to ours and go. But whatever it is, you want to send out a proposal, come, talk to us. The human subject section is a scoreable section on the grant. We're here to help. This needed some thoughtfulness. And because of the IRB structure that is maintained at Humanoid, you can do what you want to do at speed that is impossible otherwise. There was another level of thoughtfulness that went into this. How are we going to be different? People were growing organoids. We knew we didn't want to grow just organoids for the sake of growing them. We wanted to do things others weren't doing, like chronic inflammatory diseases, tissue fibrosis, diseases of immunometabolism. Nobody was doing them. You know why? It's complex. It's easy to grow cancers. They grow anyways, even if we hate them. You don't see that cancer word there, not because I don't think any little of it. Trust me, there is no disease that I hate and respect so much as that. We had a different plan for it, and I'm going to show you soon what that was. But our goals were very, very simple. Do something tough. Do something first in class. Something that has not been possible because if we did that, that's a real test. We did something. We tried something that's deemed impossible. That brings me to the most important thing that we do differently. You see, prediction in medicine is only helpful if it's, well, precise, reproducible, and relevant to endpoints that you want to see reversed as a physician in the clinic. But biology and medicine is not math and engineering, right? Maths interprets laws, physics laws. Engineering implements them with precision. Yet biology defies it. It has no rules, it has no laws. We have been told that. That was unacceptable. So thoughtfulness went into how Humanoid will accomplish this. We wanted to put it right in between multiple interdisciplinary centers so that it gets the exact kind of input that it needed. And soon you're going to see what those inputs meant in terms of speed and precision through this kind of interdisciplinary agility. So this brings me to the disclosures. Humanoids should not know what's impossible. And we made sure of that. You know how? Because I don't know how to grow organoids. I don't know even today. My lab doesn't work on it. I don't know how to code. I do not know computer science. I do not know computational biology. It was the best possible way to make sure I don't really know what's impossible, and neither do these men and women who just came and rolled up their sleeves and took a dive. Open source transparency, we publish everything. We want to make it tested by others. Oh, that's an AV failure. Let me get that right. Yes, we're not under the influence of pharma funders and lobbyists. That was important. Our motivation was really, from my side, something that I needed to use. And that is because, just this one slide, You see, G proteins, GPCRs, they guard everything you do, your taste, your smell, your mood, your bronchial tone, whether you're in love and your pupils are dilating, everything is GPCRs. It's a field that has been visited by Nobel Prize for 8 times because it's important. And here was me working around in my postdoctoral time, something fell in my lap, the proteins called GEMs. And what the GEMs did is they took away the ball from the GPCRs, the G proteins, they're hijacked. And the GEMs could go ahead and activate it from any receptor downstream. That was complex, and I needed to look at it in the way I needed to solve it in the model systems I really would believe. And that's when I called my mentors, and one of the mentors said, Pradipta, choose your model system carefully. Maybe you want to choose something you can see through it. Maybe a fish, an in vivo system. No, can't do. GEMs are not there in fish, not in worms, not in flies. I'm sorry, I got to go human. And that is when I turned into this. So it was just a need. My motivation was as a research need. And I found there was so much complexity, just like every one of you. I just could not think I would know how to use them. They were complex. They had no rigor. There was no way for me to trust what would happen. And that's it. That's what got us here. Here is where we had a chance to look at it from a different standpoint. I was trained by who is called the mother of modern cell biology, and before that, who is called the father of glycobiology. So to born to that kind of academic lineage, I only could think simple. And this is the best way of looking at simple. Nobody online or in this room would mistake any of those animals as something else, would you? Because that is Pablo Picasso, the master of simplification, the art of abstraction. Notice how each bold stroke simplifies complex things in nature. We took an inspiration out of that because we knew everybody from tech to science to anywhere else would value simplicity. Simple is harder than complex. But if we work to get there, maybe we'll be able to move mountains. That's how our Picasso was born. This one word, it puts together how we build organoid models. It is deeply rooted in the phenotype of the patient. That's what P stands for. It's informed from there, clinically abstracted into a mathematical formula, and for us to build a model and then simulate the outcomes. It's really what it says. Because art distills complexity. We scientists make simple things too complex. And so that is what Picasso is. It's the first human first framework we know of for federally mandated labs. All Picasso does is algorithmically abstracts all these cohort level information from patients, uses them to build simple models that would then hopefully, and I'll show you how reproducibly, can be used for drug discovery, precision therapeutics, so on and so on. Picasso's central philosophy is very simple. And I want all of you to simply take this if nothing else sticks to that. Patience anchor the truth. Everything else, including NAMs, must either align or be exposed as artifacts. Patients are the only thing that matters. How does Picasso work? 5 simple steps, really. Step 1 is the math. It's the mathematical abstraction using machine learning and just simple math, foundational math, finding out what drives diseases that are universal in that domain, invariant patterns. Then that's handed over to build model systems, simple, scalable, reproducible, and we do not discard animals. These animals are just elevated into vetted platforms that would be more human-like, and only those would be utilized. Then comes measure with precision. What are we chasing? What do we want to reverse? What exactly is important in phase 3, regulatory approval? Maybe we can put a measurement to that and look for whether it's going right way or the wrong way. Simulate them at population level and translate if everything else passes. Simple. Simple. Picasso codifies all of this. It makes it specific. What to look for, how to look for, what map to use. And it makes us disciplined to deliberately make things simple. Because without simplicity, there is no interpretability and translation falls through. We know that. It has happened too often. Step 1, Center for Precision comes in and uses math in order to extract the rules of biology from foundational levels of cell biology. You see, every cell in our body decides in a flip of a coin. It's always zeros and ones. Live, die, maintain stemness or differentiate, migrate or divide, regenerate or stall. You know why? Just so that we can make more complex, we can answer more complex questions, such as what's for dinner tonight. Your cells actually keep it simple. That's rule number 1, life computes in threshold. Rule 2, if decision is on threshold, then biological endurance is what patterns of invariance are. Every machine learning program, every computational program uses opposite and inverse. But guess what they leave out? They leave out these 4 other relationships. These 4 relationships tell you what biology permits or forbids. Because you see, one of the quadrants of those, that no biological samples exist. In each of these, one of those 4 quadrants are statistically low in occupancy. And those red dots are just samples in this universe. 56,000 random samples, anybody's cells, anything. And yet, you see biology forbids some places. Why? That's foundational. That's gene regulatory network. That's working at a level that biology has agreed logically is not a good place. We take that because those are always supported by feedback loop. Just like in life, our cells, if they don't get feedback, they can't move on, they can't grow, they can't learn. Feedback gives fidelity. And everything being brought down to zeros and ones makes it simple. So Center for Precision just takes a whole network of transcriptomics and turns it around and makes it zeros and ones. And one more thing, because it's zeros and ones and the network, the way it's connected, we can tap on one part and the other part has to predictably move. Those elephants playing ping pong is just simply telling you there is no other way for the network to move if one of them landed on that seesaw. It's predictable. It's certainty. Cells stay in certainty, but uncertainty, but it decides with certainty. Rule 4, disease just doesn't happen today. Disease started a decade ago. If you got diagnosed, God forbid, with a cancer today, it didn't start today. Apparently, it's 10 to 15 years ago. If you have ulcerative colitis today, guess what? It started 3 years ago. If you have Crohn's disease diagnosed today, guess what? It started 7 years ago. If you have type 2 diabetes diagnosed today, landmark work showed your beta cells started failing a decade ago. So disease doesn't happen overnight. It's a continuum. Bone extracts that continuum in math. Finally, it gives us exact signatures to trace just how much are we down the hill towards a disease? And these 5 rules make ontology-based, pathway-based frameworks completely looking for your ring under the lamp because there's light. That changes every year. That's the definition of irreproducibility. That's the definition of bias, incomplete. We rejected that. We rejected black box learning. We went with the Bond framework because it recapitulated cellular intelligence. It recapitulated logic, and we went with that. Second, we modeled them. Health and disease, anchored to population. I just told you how. But most importantly, making sure clinically relevant endpoints and therapeutic successes looked for both toxicity and efficacy simultaneously. Third, Agilent Center is right there next to Humanoid because that is how we need to make sure workflows are developed, seamlessly integrated with high rigor, high reproducibility, regulatory ready workflows. And that is possible because of sophisticated equipment that can take a pulse of the organoid every minute as we walk away for a week. I'll show you some of that. Finally, concise simulates outcome. And here I depend on mathematicians, physicists around this campus to help us out because I don't know all this. And so I'm having fun. This just happened last week. 2 mathematicians said, we want to build a digital twin of aging. Sure, how can we help? So all I'm trying to tell you is the key takeaway is model only what matters. Relevant to phase 3 approval, objectivity, rigor, and make sure patient is front and center. That said, let's see what we can do. Our first signature program is IBD, and our goal was, can we do something that the world hasn't done? We have only tackled inflammation. We have only tackled dysbiosis by giving antibiotics. Can we really repair the barrier? Because that seems to be the phase 3 success point for a drug. So what we did is we started the IBD program a long time ago in 2020, before COVID. We first built the continuum state of what health to IBD looks like. And with generous support and continuous support from our funders, we could then go ahead and train the model to what it takes to have a phase 3 success. And what would that look like in terms of gene expression without getting caught up on the gene numbers or anything. What I'm here to tell you is we could find an actionable insight. You know what it was? It was simple. It was H2S detox system in the epithelium was broken. How precise can that be? I just told you it wasn't H2S production. It wasn't H2S metabolism. It was simply its clearance. And it was 3 enzymes that Annalise rushed in to show me on her phone. Look, it works. Because it is completely out of belief that it was something like this would work out. And I wouldn't have believed it myself either. So we took that, put it on prospective cohort, built a biobank, living biobank, went ahead and did a lot of the studies, phenotype, deep phenotyping, multi-omics, tested some of the hypotheses, kept publishing. And we came to the finish line where we said, now we want to reverse what causes all drugs to fail. Guess what? 90% odds. If you don't fix this, you'll fail. And IBD has a graveyard unlike any other disease. So many billions of dollars wasted. We had biomarkers, we had therapeutic targets, high value ones, and we ran with it. This meant isogenic biobank. The organoids are from the same patients as the myofibroblasts, as PBMCs. And this is only possible because of the IBD biobank here at UC San Diego, who sees patients from all over the world. And we could dream of building complexity without losing control and interpretability. You add as you go, play Lego. See, looks like Lego, isn't it? You just snap, connect, and go. How do you make sure it works? Well, those are the broken junctions in the diseases. You know, those organoids were cultured outside the body, away from the bacteria, and they still remembered where they came from. The power of keeping epigenetic memory intact as you do disease modeling, because these junctions were broken in the disease subjects, not in the healthy. And we went ahead and showed a drug that would actually fix those junctions before and after the drug treatment. We went ahead recently and just had IND approval level studies for a drug that is licensed to a big pharma, the first biologic that reverses stem cell death in Crohn's disease, the disease that has no animal model that works right now. Look, what Ricky did is put the monoclonal antibody. This is how terrible Crohn's disease organoids look like compared to healthy, which looks very nice and beautiful round circles. We put the agonist, we could reverse all of that. And of course, as good pharmacologists would, you would put an antagonist and it sucks. Predictable. My heart is pounding. You know why? Because that happened in exactly 2 months. That it takes longer to make any drug, first of all. How about foundational science, fundamental? My graduate student said, you know, we have this crystal structure level evidence. We know this interface is important. How can we go and find a translatable methodology? So every time basic science thinks translational science is not good enough, that whole divide needs to go, because guess what? She could walk over to Humanoid, ask for a complex model of patient organoid with a macrophage co-culture model with the bacteria that comes from a Crohn's disease patient, and do reproducible, almost mathematical analysis of barrier integrity. And yet the drug was affecting the macrophage, not the barrier, not the epithelium. All I'm trying to tell you is the possibilities are endless, because we were working at some layer here that was unknown to me. This is our stromal program. Harrison Penrose, absolutely frigging good catch for us to keep him here. Look at it, what he did. What he did is he took the biobank right from Dr. Bridget Bollard, who is his commenter, built it with the help of Humanoids Director Courtney Tyndall, and identified, despite all the heterogeneity we talk about in our clinic, guess what? There are only 2 kinds of stromal cells, myofibroblasts. One that's problematic, called inflammatory. The other that's absolutely benign, called quiescent. And what he did is he made sure we were not dreaming, because the cultured myofibroblasts looked exactly as it was inside the patient. These are donor biopsies showing the biomarker for IMF is there when the QMF is not there in that patient, whereas the QMF patients, this is there but not there. See? It works. We were surprised too. And then, of course, deep biobanking, lots of phenotyping, but that doesn't get anywhere, does it? What we wanted to do is take that biomarker, take a therapeutic target, ask precision to give us the one that matters, and we ran with it. I'm not going to show you. Animal studies work. Validation in vivo works. We wanted to go in silico, and this is what matters. Dr. Boland opened up her clinic. Her clinical coordinators, Cami is here today, superstar. All of them got together, took all of that biobank information about every patient 5 years from the time they gave us their cells. 5 years. And then we did the phase 3 trial on them, just to have fun. Why not? We asked the question, should we reverse the IMF? Is that bad? Guess what? It stood the rigor of phase 3 level statistics to tell that the follow-up endoscopic score for these patients are terrible if they had IMF. Meaning they would fail everything we gave them for 5 years. And this was our opportunity to reverse that. This is a living biobank. We were working on the virtual replica of those patients for 5 years. So Dr. Sinha said that's a 35-fold risk of failure, 2.78 odds ratio that they would do bad. So Dr. Penrose went ahead and asked, does it impact stem cells? Yes. It kills the intestinal stem cells if it's IMF. He asked all of these questions, but this image is important. Notice the doctor who saw the real patient is now with the doctor who is looking at its virtual replica. And proximity of all these centers made that possible while the doctor who is in the making is watching. It's important. You know why it's important? Because it's a human connection. And we could build it only because of the proximity. And this is the complexity we can go with. You want to measure barrier? Measure it. Measure it for what it means for phase 3 regulatory approval. Let's look at it. How about we measure it with automation? We have that. How about while we do automation, have high throughput level of 96 well format, one patient a well, 96 wells for all I care. Can we do it? Yes, we can. You're looking at IMF being reversed by a doctor who hasn't even been recruited as our faculty force. Don't we want him? Harrison, you'll pay me up for this. But that's not enough. That same experiment gave us 3 more readouts. And that's what I mean by workflows. You want rigor? This is rigor. We said we have to set the standard that way because those supernatants, those cells went farther because those patients gave us trusting we would use them well. And then the superstar who is waiting to get into medical school here, he threw those IMF supernatants on macrophages. And look what happened. Those macrophages couldn't clear the bug. Those macrophages sat with the bug. And that was how it mirrored one of the most classic features of IBD, which is difficulty in clearing bacteria. So all I'm trying to tell you is we have a slew of drugs that can work on stromal disease in IBD. Never thought of before as possible because this biobank is unique and the only one in the world to have it with stable phenotypes. Cancer. It consumed my biological and my academic mother. I told you we had a different plan for it. We wanted to do something that was not possible because nothing today available as treatment can take care of a cancer stem cell. You know why? Because we don't know what they are. We don't know their identity. You can take an LGR5, which is just a marker, you can wipe it out, and we know it comes back. The cells come back because it's not about LGR5, is it? We took the mathematical approach. This just came out. UC San Diego, thank you, Media Communications, for running this. We put together a pipeline that can work for any cancer. All you need is our goal was simple. Find the cancer stem cell in whatever tumor, differentiate the hell out of it, and then if it decides to die, it dies. And that's what we did. It needs simple things. It needs a lineage determinant, one that would have the ability to predict outcome. And that's all. Simple. Simple. Allowed us to build a target, build a network, pick a target, do the validation. And here is what you're seeing. Before treatment, they did not know they belonged to colon. But after treatment, all of these brown dots are telling you they remember what it is to become a colon again. This is before treatment. They had signaling like cancer cells should. Nuclear beta-catenin, that's bad. After treatment, you can see they remembered how to differentiate. You can see those junctions. What we did is we were able to find a drug that did not even touch the normal stem cells, did not touch the normal differentiated cells, did not touch the differentiated cancer cells, but selectively, also selectively just destroyed the cancer stem cells. And it didn't do anything to the healthy. Turns out it was a pathway discovered in my lab in 2015, when I had no clue who would be doing this a decade ago. This is the picture of a cancer stem cell before it commits suicide. You see that red chicken wire? That is the pathway. That pathway, once triggered, makes that cell go, oops, I don't want to differentiate. I don't remember how that is. And now it's programmed to die. But can simple move mountains? Apparently, on a cohort of 2,000 patients or more with 10 separate independent cohorts, Dr. Sinha would go on to show that if this drug were to go into market, it could save relapse and death by 50%. But can simple move mountains? Turns out there are numerous cancers that go through intestinal pathway to become tumor. You're looking at stomach cancer, same for esophagus. Guess what? The drug works. It tends to differentiate those cancers. Here is organoid, here is organoid, here is animal xenograft. Before and after. This gets me to the second most robust program we have, lung fibrosis. Can lung fibrosis be flipped to regenerate? Let's try it. What is the deadliest disease? Worse than cancer. 3-year survival if you have progressive idiopathic pulmonary fibrosis. Nobody knows what it's caused by. That's why the word idiopathic. But that's not because we don't know what it causes, what it's caused by. The cell that we really wanted to kill was a DATP. It's an alphabet soup, but it stands for damage-associated transient progenitors. We know who they are, we know how to track them, we know how to look for them, we know exactly when they emerge. You know what? We know that they drive fibrosis, but guess what? We don't know how to model them. That's why we can't screen for drugs. That's why we can't impact human drug discovery, because everything that worked in mouse and model of lung fibrosis hasn't translated, has mostly did. We were lucky because bang in the middle of COVID, Humanoid delivered world's first adult stem cell model of lung, all 5 epithelial cell types. The FDA got excited about it, reached out to us. We served from Stanford to Berkeley to coast to help everybody run their COVID programs. We published with them, we helped them out. Right now, many other places are asking us for help with this model. This is why this model works. It's called proximal area, distal area. It represents the entire lung. That meant it had the stem cells, the progenitor cells. That gave us hope. We could model fibrosis. We went the route of abstraction. We put bleomycin, which is a common thing used to model fibrosis, and guess what? Just like in men and women, men smokers, higher risk, you see, every time you see green and red, that's DATP. So those arrows are DATPs. Remember, DATP bad. DATP bad, damn it, bad, fibrosis, right? We know females don't get it, and turns out organoids told the story. We didn't see females get it. We were encouraged. But that's when we went to Precision and said, tell us what it takes to make DATPs. And Precision looked through 46,000 human samples, all on the internet, anybody's samples. And it told us, it's simple. You need 2 hits. You need hit 1, hit 2. This is being patented, abstract submitted. I'm not going to be able to tell you the exact chemicals because of lots of online presence today, but it told us how to get them. And we did. You want to see how it looks? This is control. This is our good old bleomycin. And this in the middle is Picasso. Every yellow is a DATP. Red and green together is a DATP. We made them, but can simple move mountains? Let's see. Turns out Ella and Courtney working together took away hit 2. That became our signature drug. It can take away DATPs or its ability to live. If it doesn't live, it cannot fibrose. But we weren't happy with just that. We said there's another disease that looks just like IPF, has fibrosis just like IPF, male smokers, not female, blah, blah. We went to Dr. Boss and we said, help us out. And he helped radiate, zapped it out, and within a week, we had what was a radiation-induced pulmonary fibrosis, the first human model for RIPF. And I'm telling you from last week's microscope data that the inhibitor for HIT2 took this out. So now it works to drug across pulmonary fibrosis. But that's not enough. The team went and asked, can we harness regeneration to reverse fibrosis? And this is where I don't have the time to show you, but I'm going to show you something funny. Turns out this is what we learned. All alveolar cells have junctions. And the tight junctions, if they're too tight, you have fibrosis. Too loose, you have barrier gone. You're flooded with water, you can't breathe. Just right. Like in life, just right. And just like in life, you need only two tools to get by. A little WD-40 if it's too tight, a little duct tape if it's too loose. Guess what? They made a molecular WD-40 and they are ready to go regenerate the hell out of it to make lung go from fibrosis to regenerate and back to normal. And how did that happen? Again, with precision. We knew what to target, exactly the sequence to target, exact protein to target. We just went with it. And this is just another way. These are alveoli filled with air. I have never, in the last 4 years of Courtney showing me lung organoids, have I ever seen lung filled with air like that, the air sacs. For me, this is proof that there's some things can be done differently to make it better. Picasso's last part, which is, I think you all will like it. I've struggled with weight. Which one of us doesn't want to get rid of it? So we asked the one question that hasn't been asked in metabolic syndrome. If the problem is too much fat, can we remove it? Turns out the body's only path to remove it is RCT, called reverse cholesterol transport. We know who they are. Macrophages must take it out of the tissue and push it out. So we did what we do, which is math, came out and prioritized what to target, and we could get the fat out. You're seeing those green blobs go away. That's defatting. But that's not enough. We went ahead and looked at the mechanism. It was a specific structure-based, pathway-based, and Dr. Katkar asked a simple question. Can we move mountains? Recruited the patient, built plaques in a dish, did the entire thing with human donors, figured out how to build it, and here's the data. You see, if you have ongoing injury and you add any of the drugs in the market, they all can stop the formation of these fatty cells, which is this lambs, which are filled with fat. But what if you were to remove the fat? None of the current drugs work, only the new drug works. This told us we have a new way to actually deal with defatting. And by the way, while at it, he could show the liver gets rid of fat, the adipose tissue gets rid of fat, the plaques in your artery get rid of fat. But we asked, will this change survival? Will this change acute coronary diseases? And the answer is yes. For each molecular signature, we simulated what the impact would be. And turns out if this drug were to go into market, it would give a protection from the risk of heart attack by that much. Odds ratio 0.012. That's a molecular score. So I'll just end by saying we have a lot that is going on. Questions being asked about multicellular life, questions being asked in cancer prevention with hereditary diseases, questions being asked, can we flip the coin on cardiomyopathies that are rare diseases where junctions are ripped apart? And we have programs that are addressing it, even if it's N=1 trial. That is where things are headed. We have new immunotherapies in development, 4 programs, one of them in upper gastrointestinal cancers. We have very aspirational programs. That's about charting the metabolite atlases of the longest communication between two humans. That's a mother and a child, in utero and after birth. And we have lots of things to think about when it comes to development, autism. And we're working on all of those with lots of people here on this campus. I want to end by saying in 2008, Declan Butler drew this picture in Nature. Things fail because we lack those 3 R's. This is my little version of it, so I apologize, but sure, that's how it looks. This is what we propose. Picasso should be able to be a sturdier bridge. It doesn't eliminate people's animals. I do not think that's necessary. We can just make humanness objectively better. That's what's possible. With that, thank you very much.
Thank you, Pradipta. In case you missed it, enthusiasm and energy is a major ingredient in being a serial academic entrepreneur. So we have 5 minutes before we have to get out. So quick question in the audience.
Where it fails, it can't even get.
Let me make sure I understand your question. Is it that it works in organoid, doesn't work in human, or are you saying it penetrates accessibility to in the organoid and not accessibility? Accessibility is your question, or efficacy?
Just accessible.
Oh, accessibility of a drug. Great question. Let me quickly step back a minute. Why do drugs fail? 55 times is efficacy failure. Toxicity unmanageable is about 19-20%. If the target was good, you can see the accessibility of the drug, how good it gets to it, the potency of its engagement with target, all of those factors that we can't work on, we don't work on. Those factors are much better off if the target was great to begin with. If it was expressed in the right place, right time, with the right cell type. So yes, it's possible that accessibility and potency of a molecule is 100% important, but nothing is as important as picking that target, which arguably is step 1. Do you see my point? Thank you.
Okay, more questions? Anyone? Anyone online?
Oh.
I see it here. No. This is a question online. Does Humanoid plan to incorporate organoid in a chip? Is Humanoid also focusing on infectious diseases?
Yes. Just to let you know, I recently had we have in the past had the fortune of working with a big chip maker here in the United States. I'm not sure if I'm allowed to tell the name, but I also just came back from a conference in Taiwan where, again, chips are really, really big time favorite. There are some things we cannot do here. A blood-brain barrier needs flow. An intestinal peristalsis model needs stretch. I do not want to leave anybody here with the assumption that simple means sacrificing. Simple means if it's needed, we will only use it for that purpose. So I'm pleased to tell you that there are flow-based methodologies that Director Tyndall is looking into where we need it for the blood-brain barrier of the developing, maybe, if we were to study that transporter interface, and we will someday go into the intestinal peristaltic stretch.
Last questions? Got 1 minute. Right, I'm sure Pradipta is available for more discussion afterwards.
I am. Thank you all.
Thank you all for coming, and we have to get out.
Thank you. Thank you.